From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded at the American Society for Microbiology meetings in Los Angeles, California on June 22nd, 2025. Hello, Micronauts. Can you say hi back? And welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today, friends and Quality Quorum folks, is the 99th episode of Matters Microbial. I am so very grateful for every listen, view, and email. Thank you for spreading the good microbial word. Every time that I teach microbiology, I work hard to give a historical perspective. Pasteur, Koch, Semmelweis, C.B. Van Niel, Woese. The list is long, and some students aren't as excited about that as they might be. But I contend that learning from the past informs the future. And it so happens I attended a session just a few minutes ago that really turned my microbial head all the way around. A few years ago, I traveled to Delft in the Netherlands and was able to think about the history of microbiology. One name, and it was the subject of this session, incidentally, that should be better known is Albert van Kluyver, 1888 to 1956. And actually, it's from him that the famous quote about E. coli in elephants originated. He worked hard to promote the concept that metabolic events were all about electron transfer and the unity of biochemistry for all living things. So I cannot express how delighted I am to have Dr. Dianne Newman, the Binder and Amgen Professor of Biology and Geobiology at the California Institute of Technology in California, to this podcast. Welcome to the Quality Quorum, Dianne. It's wonderful to see you in person.
Thank you very much. I am a big fan of you and your podcast, and I will be very honest. I thought we were going to be in a little cubbyhole, not in front of everyone at ASM. And so I'm going to channel my inner microbe and try to adapt.
Well, you know, it's all about a community, isn't it? That's one thing we're talking about.
Yeah.
And I think that's part of it. As I mentioned, I just came back from this wonderful session. One of the issues that I've had, Dianne, is I've taught a fair amount about the unity of things and at the same time the diversity of microbial life, but I never saw it so clearly put as yourself. Can you give everyone kind of a thumbnail about what inspired you to put this session together?
Yes, multiple things. So I have been teaching microbial metabolism now for 25 years at Caltech. And I begin my class with this paper from Kluyver, this 1924 paper, which is entitled Unity and Diversity in the Metabolism of Microorganisms. And we have a 10-week class. The students read this paper in preparation for the first day of the class. And then at the end of the 10 weeks, they read it again. And what I find is that I personally get something different from this paper every time I read it. But for my students, what's really wonderful is that in the course of really learning the crux of catabolism, that's the focus of my class, they sort of progress in a spiral back to the paper where they go up in an understanding and they have a really, I think, much deeper appreciation for what Kluyver was describing. But more importantly than that is what he says itself in this wonderful paper. He calls for looking for unity amidst diversity, which I think intellectually is something really profound and something that we all should aspire to. I think when we attempt to stretch ourselves intellectually in this way, it's when we stumble upon the principles that are really important in science. And so I like that myself as a goal in the research that I try to do is to try to find, higher principles that transcend the specifics of what we're doing. And so I love this paper because I think it really inspires that. And this is why I love to talk about the paper and why I decided it was really important to celebrate the paper in having a session here at ASM.
Well, it was a very important session for me because I'm getting set to teach microbiology for the last time, ostensibly. And I like to come back to the sorts of things that you're discussing here. I really like the approach of exposing students. Exposing is not a good word in microbiology, is it? Exposing students to a particular set of concepts and then come back at the end of the course to see how it's changed the way they think. And I've done that in lots of small ways, but I am going to be putting a link in the show notes to this particular paper so you all can experience it. I mean, I spent perhaps 5 minutes on Kluyver when the last time I taught micro, and I'm going to be spending more time, and I'm stealing photographs from the session for it.
Fantastic. That's really great. Well, when I first read this paper, I was just in my very first year as an assistant professor, and I was reading a paper a day with my first student, Mario Hernandez. And I remember when I read this paper and I got to the very end, and he talks about this concept of a manifest unity that emerges from all of these patterns that one sees. I literally got tears in my eyes. It really hit me at a very emotional level as well as an intellectual level. And I'll be very honest, I think another reason I was excited to hold this session today at this 2025 ASM is because I think that message of unity and diversity, not unity or diversity, but rather unity and diversity, is more resonant today than ever before, in a transcendent way, not only about science, but about the process of doing science and many other things. We can leave it at that.
And I'm just going to say parenthetically that one of my very first undergraduate research students, Dr. Paula Wielander, presented and put exactly that point out. And it's a good one to remember. And the microbes do have a lot to teach us, don't they?
They absolutely do. They were first in, they will be the last out.
It's funny, I've been saying first evolved, last extinct. That's what Abigail Salyers and I used to say back and forth.
Yeah, I probably picked it up then too when I was a student in the microbial diversity course, which is nothing but an extension of Kluyver's legacy here in the United States. From teaching Van Niel, who started the Pacific Grove course, to then the legacy that continued on into the microbial diversity course at Woods Hole.
I find it so exciting because when I took this course, and I was trained as a geneticist and I loved microbes, but I didn't have an undergraduate degree in microbiology, but I loved it and I was an autodidact about it. I was fortunate enough to get into the microbial diversity course, and there's C.B. Van Niel's notebooks.
Yeah, wow.
And there's the history of every student who had been involved with that.
Well, it was Holger Janisch who started that course, and Holger, I believe, took the class from C.B. Van Niel at Pacific Grove.
And when I spent time talking to Esther Letterberg, she talked a lot about C.B. Van Niel as well. But the point that I'm trying to get at is that got me thinking about historical issues, because I kept seeing things in the notes that I thought were modern ideas of mine that people had thought about in the 1920s and 30s.
Oh, yeah. I tell my students, I think we all recognize that there's very seldom, if ever, an idea someone hasn't already had. It's more a question of their capacity at the time with the technologies that existed in the day to pursue it to its fullest extent. And so we would all do very well to go back and mine the literature of the 1920s and 30s, where people had to really use their brains more than whatever random technology they wanted to throw at a question. The elegance and the clarity of how they posed questions and the rigor of the logic where they pursued them is deeply inspiring. So that's a huge source of inspiration for me personally.
It's before your time, but there was a wonderful paper called Logic and Toothpicks.
Yeah,
it's
not before my time. That's about Jonathan Beckwith, right?
That's correct.
That you could answer no question. You needed nothing more than toothpicks and logic to go after a good question using bacterial genetics.
Well, I mean, the most expensive and best piece of equipment we have is here, right?
Absolutely. And we all have to resist ChatGPT destroying our ability to use our brains to their fullest extent.
So, I was fortunate to go to Delft a few years ago, and Leslie Robertson was kind enough to show me around the Delft Museum, which has since closed. I got to sit at Kluyver's desk.
Oh, wow. Lucky you.
And I was just you're all used to this if you know me. I was prancing around a bunch because I was so happy with everything. And I saw C.B. Van Niel's notebook with perfect penmanship. Irritating. And then I saw Beyering's notebook just like mine, including a circled diagram saying sporen. And so it's so interesting to see that historical precedent, and it's come back. And this is important to remember, and that's what I really want to hear from Dianne, is about how this has really informed the different ways that we look at metabolism. And as you've heard, I'm sorry to keep, I've just been proselytized to, so now I'm passing it on to you, that this idea of unity and diversity being so important. So Kluyver did what that was really exciting?
Well, I think more than anything, he really thought deeply about what other people had done as much as contributed his own original research. He did both. And I think that synthesis is sometimes underrated. I think that there's so much we can gain by really deeply thinking about what other people have already discovered and how it connects to other facts, and then try to thread between them. I think that that is a very creative act, the act of unifying information, even if one wasn't the person who, got that individual bit of information. The creativity comes in connecting it. And so I think he was an absolute master of doing that. And another, I think, extraordinary contribution he made was being bold. This was what my student John Semniecki noted in our symposium. He had the boldness to say, we can do better. We have some evidence of things, but we don't have an explanation for them. And yet there must be a way to bring it all under a conceptual framework. And I think also in science, people who have the boldness to put out a clarion call for others to step up are people who really advance the field. And so I think that that was a gigantic contribution he did that then inspired a lot of the most seminal work that followed, that led to the discoveries of the principles underpinning bioenergetics and the electron transport chain, and the principles underpinning central metabolism. I think really Kluyver was one of the people who catalyzed the conceptual frameworks that then followed.
Without necessarily knowing the pathways. That's the part that's interesting about it.
That's right. Yeah, in his paper, in the 1924 paper, he points out sort of different precursors, different metabolites that we now recognize as being intermediates, for instance, in the TCA pathway. He was seeing in diverse organisms them popping up under different conditions, and he intuited that this must be because there must be conserved pathways in each of these organisms, but that when they were grown in slightly different conditions, it led them to favor, excretion of one intermediate more than another. And so I think this concept is also very profound, that the things that we observe about an organism are very conditionally dependent. And that's something that Paula Wielander, who spoke in the session, very beautifully emphasized in her talk and has really mined a lot in her own research.
I couldn't agree more. And when most people are exposed in textbooks to Kluyver's work, they're mostly about the idea, I'm going to simplify it by calling it redox, and it's not as simple as that. The idea of electron donors and acceptors, and all of that's important. And then he's not given the amount of credit that Monod got for the, if it's true for elephants, it's true for E. coli. But in fact, what Kluyver said was, do you happen to recall?
Yes, from the butyric acid bacterium to the elephant, it is all the same.
It is all the same. And I want to emphasize this idea because something that I think we don't think about here, many people when they look at microbes think that they're fundamentally different. And they're using the same set of tools because we are descended from them. Makes sense, right? And this idea, I mean, look at the fact that people didn't call cyanobacteria that. I still find people who say blue-green algae. They're not algae and they're not blue-green. So in any event, what I'm trying to say is that idea of unity is something that can be carried too far. And I do that sometimes, right? Or we can go too far. It's like the lumper and splitter kind of idea in taxonomics, where you can carry it one way or the other. And what I particularly appreciated about the presentation and the paper is that you can have both.
Right, exactly.
It's like the meme where the little girl's saying, why not both?
Yeah. Yeah, no, that's what, and for me, that's what makes it truly beautiful, is that there is an overarching explanation, a principle that then is manifest in different instances, different examples. So, for example, my favorite, if this is the electron transport chain, right? The concept of oxidative phosphorylation is a beautiful concept, but it can be achieved with very different, things, substrates that feed into the chain and that accept the electrons at the end of the chain. But yet what's happening really is all the same thing ultimately at the end of the day. It's proton transfer coupled, proton translocation coupled to electron transfer, or sodium transport, right? You substitute the cation, and the electrons, are coupled to it, and what they go from X to Y can be different, but everything else, it's the same general idea. And I think that's really elegant.
One thing that I've talked about a lot are what I call the deadly centrisms in the way that introductory biology is taught. And of course, eukaryocentrism is the first and most deadly of all of them, because that's how it's taught. It shouldn't be, but that's how it's taught. Oxycentrism deals directly with what you're saying. All the emphasis is on electron transport dumping onto oxygen. And when you show the pairs of acceptors and donors that you can have in the microbial world, minds are blown.
Right.
Especially when you look at the tiny little differences. And I'll say, you go look in an anaerobic pond and you'll see all kinds of things bopping around. They're not making 32 to 36 ATP per input glucose, but they're still doing just fine. And that's the way the whole world was for 2.5 billion years.
Amen.
Yeah.
That's right.
I proselytize, but it's true. And so one of the things that I wish we could do is change the way that introductory biology is taught, just for that reason. And I get called an evangelist and a centrist and all kinds of things, and I am.
I would share your enthusiasm for changing the way we teach introductory biology. I agree with that very much, and I have a dear friend, Margaret McFall-Nye, who has been leading the charge to do this, and I had the privilege of teaching with her, actually, at Caltech, where we attempted to do just that in our courses, where I would begin introducing a concept, a principle that was first established in microbes, and then she would extend it to show how it was relevant for mammalian systems. And this was an incredible experience where I think we were all enriched by seeing how these ideas cross-cut between organisms, that there was this underlying conceptual unity, and yet it manifested in ways that were superficially so diverse, but yet, truth be told, they were very much instances of the same thing. And I think that is a moving concept, and also intellectually pretty cool.
Putting a phosphate on something, taking it away, moving a solute across a membrane, you have differentials in charge. All these things are true for all that lives at this point.
Yep, that's right. I mean, life has to exist. It has to persist. It has to co-evolve with its environment. It has different ways to achieve these things. I think what you were saying earlier about maybe molecular and cellular biology focusing a lot on the central dogma, and people somehow thinking of metabolism as something less interesting, less fundamental. Well, of course not. I mean, all of the process of replication, what we need to do to preserve the integrity of the genome, to replicate it, requires energy. And that energy has to be conserved. And that's where metabolism comes in. So there is no part of the cell, whether it's separating the inside from the outside via lipids or, copying its genome, that doesn't interface directly with metabolism, which brings us right back to Kluyver. It's a system, and systems biology in its fullest extent, I think, helps us really understand the interconnectivity between all of these various elements of the life sciences that tend to become a little bit pigeonholed. I think we're now in a time where we can really transcend that and really see the way in which they're connected so deeply, and try to figure out actually how they interact, these different functions of the cell, in a way that's really rich, whether it's in a microbe or in a mammalian cell.
Well, I remember being a student, and when I was learning basic metabolism, I had my 3x5 cards, and that's I wouldn't say it's underwhelming, it's whelming. And I would memorize things, but what taught me to understand metabolism is the Krebs cycle. because it was so interactive with everything else. And that takes us back to the diversity and the unity idea.
Right.
And it's something, and by the way, and I'm looking at Krista right now, who suffered through my classes all these years ago. And if I were to ask Krista what the most important enzyme on earth is, I love that question. And don't worry, Krista, I'm not going to put you on the spot. Many students talk, as you mentioned with Central Dogma, about, DNA polymerase or ligase. And I'll say, well, how about Rubisco or other enzymes that fix carbon? What about nitrogenase? And here's the thing. They don't think about the fact that an autotroph has to build everything from CO2, and they don't appreciate the level of nitrogen. So this takes us back to the universality of life, but we do get blinded or blindsided or whatever you want to say in the way that we look at things. And this is, I think, the value of Kluyver's approach, because he knew very little compared to today, but he was clear in seeing how it was all related.
Yeah, I mean, I'll be honest. I absolutely had a distaste for memorizing the intermediates of the Krebs cycle. I'm not even sure I could do it right now if you put me on the spot. It was not the type of thinking that appealed to me. I found it horribly detailed and really boring. And apologies to people who love metabolic pathways. I've grown to appreciate them. But what I loved about Kluyver was sort of the permission to think a little bit more abstractly and recognize that those were important details, but yet they were details. And that what was, at least for me, a lot more interesting was how you could generalize and understand really what those details were enabling the cell to do. And so when I teach this, I try to abstract away from those metabolic maps made by, Roche. You get those amazing posters. I have one in my lab.
I remember. Yeah.
But I think it's actually much more intellectually compelling to say, no, that is important and has its place, but we can actually distill the bigger picture concepts here a lot more simply. And at least for my mind, it's easier for me to retain information at that level.
Where they connect
Correct.
up. So I'll never forget succinyl-CoA, because I know it gets taken to make things like hemoglobin, yes, and chlorophyll. You don't believe it, but I'm actually quite nervous right now. But you don't believe it.
Amy Vollmer really did me a solid once, because she came to my class and she brought these wonderful slides. She gave a lecture that I would be right with you. Don't feel embarrassed. I would have forgotten too exactly what succinyl-CoA goes to, but Amy wouldn't have, because what Amy did was really bring one of these wonderful maps of the TCA cycle and then show how off of it bled pathways that led to the key, precursors we need to make all of the important constituents of the cell. And that this idea that TCA is just about oxidizing carbon to feed into the electron transport chain, well, sure, that's one thing it does, but it's intimately coupled to anabolism by generating the precursors for all the other things we need. And so I think that that level of retention of information is what is most important.
And it's the connections, folks. That's the important thing. So my students have gotten to the point where they're tolerant of me after all these years with this, but they really don't like the individual pathways very much. And I keep saying, what's the microbial perspective? And yes, I know they don't have a perspective, or do they? But that's really important to do. When I tell them you can run Krebs cycle backwards, they're disturbed.
Well, that's the beauty of biochemistry, is that it's reversible, most enzymes.
That's right.
But I guess I've come to believe that there is no one right way or right level of resolution that, as a scientist, one ought to operate at. I think it's a very personal thing. And I have full respect for people who want to drill deeply into, for instance, structural biology, or even beyond that, into understanding the bioinorganic chemistry of a cofactor. And even beyond that, to like the physical chemistry that allows that cofactor to do what it does. And then beyond that, to the mathematics that underpins the physics. I think we all have our level of abstraction that we enjoy, and hallelujah for that, because there's a lot of different things to do in science. And so we each need to find our own niche, and there are a lot of niches.
I don't know if you'll agree with this, but coming back to the idea of unity and diversity, there's a lot that goes into the microbial sciences today, as you just mentioned. And so just as sometimes we need a big tent for different ideas, we need a big petri dish for our field.
Yeah.
And it's true, because there's nothing wrong with loving the mathematics of it. I'm married to a mathematician, so I hear about this all the time. And so in other words, if I can't do the mathematics, and I can't, that's okay, because there are other things I can do, and we're better working together, which brings us back to this unity and diversity idea.
Absolutely. Bingo.
So we have a relatively short session today, but I'd like to spend a couple of minutes for you sharing with the audience how you got to be a microbial scientist.
Okay. It certainly was not something that I ever expected I would be. I was a German studies major in college, but I'm very embarrassed to say I didn't like biology at all. I didn't take a single biology class as an undergraduate, but I did like physics and chemistry. And my father wasn't thrilled that I was going to be a German studies major because he was worried about my job, my ability to get a job, which was a very reasonable thing for a parent to worry about. And so I went to graduate school to get an engineering degree in environmental engineering. And my first semester there, I took a class on environmental microbiology with Lee Krumholtz, where I had to memorize the TCA cycle, and I wasn't going to do it. And the only reason I passed the class was my dear friend Jenny Jay, who's a professor at UCLA. I think she's here at this conference today. She forced me to memorize it for the test, and I promptly forgot it. But what I did get from that class was an appreciation for the chemistry the microbes catalyzed that was really important. And this was in an environmental engineering department where back in those days, this was in the early 90s, Perry McCarty at Stanford was doing all this great work on PCE degradation. And I was learning how in groundwater systems, microbes could eat toxins and mineralize them to CO2. And I thought that was just phenomenal. And so that's what grabbed my attention. And I was fortunate, extraordinarily fortunate, to have a mentor who, although he studied marine phytoplankton, he was generous enough to allow me to study bacteria in his lab. And then, because I hadn't had a background in microbiology, I took the class with Ed Ledbetter and Albert Saliers at the at the MBL, Marine Biological Lab, and was exposed to Kluyver there in that class, and to the Delft School, and the rest is history. That's how I got in.
So one of the things I talk about in the podcast a lot is there's no one path to become a microbial scientist. And a lot of students that I encounter think that, everybody was always of a particular type with particular interests. And the people that fell in love with microbes at the age of 10, like myself, are relatively few and far between. There are many paths. And again, diversity and unity.
Absolutely. Well said.
So I don't know if anyone has a question. I'm watching the time carefully, but I think it'd be a great opportunity, because how often you get a force of nature like Dr. Newman here?
I just wanted to reference the Van Niel's notes, and his handwriting is on display in the archives exhibit over here. We have a little touchpad interactive, and we touched on his legacy of pedagogy, connecting with the Delft School as well. Thank you for your comments. I found them to be very appropriate and very timely right now, both within science and within our larger experience, I think. So there's a lot to be gotten out of that. Thank you.
Thank you.
So I wanted to say exactly what you heard there. There's the wonderful exhibits of the history of microbiology, so worth your time. And I'm looking at them, and I don't know if you know this, but when I was at the Delft School, I saw one of the lyophils that Behring had of Photobacterium that was broken open, and I said, "Can I have that?" And Leslie said, "No." So I always come back to what Indiana Jones said: it belongs in a museum. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode with, as always, epic links will be available at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, And you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. The very impressive Dr. Dianne Newman can be found in the Division of Biology and Biological Engineering at the California Institute of Technology in Pasadena, California. Many thanks to Ray Ortega and David Renata for superb editing, and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our live Quality Quorum today. See you next time on Matters Microbial.